So firstly, just a very quick introduction, which I'm sure you actually don't need. You all know what Verticillium wilt is, otherwise it probably wouldn't be on today. But it's caused by the soil-borne fungal pathogen Verticillium dahlia. And once introduced to a field or farm, it's virtually impossible to eradicate. It survives in the soil for a very, very long time, so reports of at least 14 years. And that's why we talk about controlling the build-up to stop the build-up of inoculum, because if you are planting a host and it's building up with every time a host is grown, you can end up with very large yield losses, and fields will eventually become unsuitable for cotton because of the ever-increasing inoculum.
See?
So reports of estimate losses.
I'm going to text you his contact. All right.
Okay. Is that okay? I can just hear people. I'll keep going. Reports of estimate yield losses range from 10 to 62%, we've been told, and pretty well just, it's really focused on eliminating microsclerotia. So everything we do is focused on reducing microsclerotia in the soil. And that's why an integrated approach is absolutely essential, and I'll talk about some of those points. I've asked to talk a little bit about some of the terminology which might be confusing or unfamiliar. So I'll just run through this. So what is a pathogen? Well, it's a microorganism that can cause damage on a host. So this is Verticillium dahlia, the spores, and this is disease. So a pathogen is any microorganism that can cause disease on a host. So what is a species? So we are Homo sapiens. The genus is Homo and species is sapiens. All living things are explained in this Latin binomial. So, for example, Verticillium wilt caused by Verticillium dahlia, the genus is Verticillium and dahlia is the species. But there are many other species, not just dahlia. So here we have the genetic structure where we have dahlia here, and so we can characterise the species based on phenotypic and genetic data. So I have dahlia here, and then we have all these other ones. So that hopefully just explains what we mean when we talk about the species, Verticillium dahlia species. An isolate. So basically, when we recover a fungus from an infected plant tissue, that is, the isolate is what we've recovered. It's this fungal culture. Microsclerotia. So this is the most important part of Verticillium disease. It's the resting structure. And this is the long-term survival structure that I mentioned before. A propagule. So it's any portion of a fungus, such as a spore or microsclerotia, from which a new individual can develop. So when you might see a measurement of propagules per gram of soil, such as ppg, it's something it's a portion of that fungus that's been measured in the soil so that we can quantify that pathogen in the soil. Inoculum is any part of the pathogen that can initiate infection, and I will be talking about the life cycle of Verticillium dahlia, and it starts in the microsclerotia germinating. So this is what we would term the microsclerotia would be inoculum. Pathotypes. So mention the genus and species, and the species is dahlia, but we can further divide the pathogen into these pathotypes. And the classification is a way of distinguishing it from others based on the pathogenicity on a specific host. So We currently know we have two pathotypes, the defoliating and non-defoliating. So it's really just it's just a way of distinguishing some groups that are genetically distinct but are still within the same species. Virulence. This is the degree to which that pathogenic organism can cause disease. And we often use virulence and pathogenicity interchangeably. So I don't know if others may see there's a difference, but I really feel virulence and pathogenicity are basically the same. And yeah, as I said, it's the degree to which that organism can cause disease. Now, thresholds. This is related to the inoculum density, and it's the amount in the soil required for disease to develop. So that would be a threshold inoculum density. But it's really important to remember that the level of infection, despite the quantity in the soil, that level of infection that you're going to see is highly dependent on the environmental conditions. VCG, this stands for Vegetative Compatibility Group. And you may be familiar when we talk about Fusarium VCGs, but this also can be done with Verticillium. And basically, it's just a way of grouping genetically similar isolates from to separate them out. And so we have VCG1A, which is the defoliating. We have 2A, and this is, so internationally, these isolates are termed non-defoliating, but in Australia, we know that some of our 2A do defoliate, and quite significantly. And then VCG4B is non-defoliating, and that's that mild strain that we originally was detected quite some time ago in our cotton. Symptomatic hosts. So is this always an ever-changing area, whether something is a symptomatic host or not, because Verticillium dahlia has this incredible ability to evolve to become pathogenic on new hosts. So a symptomatic host is a plant species that is infected and then displays symptoms of infection, such as cotton. If it's a non-host, it actually doesn't harbour that pathogen. So that pathogen, you will not be able to isolate it out of the vascular tissue. And some of those crops are grain sorghum and forage, but also some millet varieties. But an asymptomatic host, it harbours the pathogen, but there aren't disease symptoms. And some of these are still very good options for rotations because the amount of the level at which the pathogen is invading that host is quite minimal. So we know, for example, that maize, corn is a really useful crop in a rotation to reduce disease and reduce the microsclerotia, even though it is termed an asymptomatic host. So now on to the life cycle. So the life cycle starts when the cotton plant emerges. This is the microsclerotia, and it is going to germinate by stimulation of the root exudates. Then that pathogen will then grow and penetrate the roots. It colonises the root cortex, and then it moves into a parasitic phase and colonises the xylem. The xylem in the plant is the transport organs for water and for nutrients coming from the soil into the roots, up the stem and into the leaves. So this whole area is going to be colonised by the pathogen. And because of this colonisation, the plant's response tries to block that colonisation, and that's why you get plant wilting. because there's all these tyloses and gels and gums being produced by the plant to say, hey, I'm trying to stop you invading me. So that's when the plants start to wilt and you get chlorosis and necrosis as the pathogen starts to colonise. Eventually then you start getting death. And in this phase, it's called saprophytic phase because the colonisation all through the dead tissues, then you get the development of the Microsclerotia. And this is this is the area that we're trying to prevent. We're trying to prevent the development of Microsclerotia. We're trying to stop Microsclerotia, as many being brought back into the soil, and we're trying to stop then this Microsclerotia surviving in the soil. Because as I mentioned, this can survive for a very, very long time. So how does disease, like what do you need for disease to actually even develop? So this is called the disease pyramid, and I'm sure many of you have heard of this because we do talk about this quite a bit. And it's the three things that you need for there to be development of disease. You need the host, you need a pathogen, and you need a conducive environment. But in addition to this, to get extensive disease, to get a disease, an endemic and widespread disease, you need these two things as well. You need time and humans. So firstly, I'll just touch on the host. Now, CSD, and you'll know, have done a tremendous job, and with the breeders, of course, and then ranking all of the varieties that they're producing with the highest V rank being the one that, or the highest V rank that's most suitable for your region and your cropping area must be planted if we're trying, to manage this disease, because the higher the V rank, the less disease, the less infection, and then the less Microsclerotia are going to be put back into your field. Now, I mentioned about the pathogen and the different pathotypes and the VCGs, but another factor that is really difficult about this pathogen is that we're seeing also different virulence or pathogenicity of isolates in fields. So you can have the same inoculum level, the same environmental conditions, and this was a glasshouse experiment. So we've got the same level of humidity, the temperature, and inoculum level, but we've got different VCGs down here, but we've got actual different levels of disease developing. And that is because the isolates differed in their virulence. The environment, and this is obviously a huge influence on the development of disease. One of the concerns, obviously, is that our varieties are temperature sensitive. And so you might have a high V rank variety that has good resistance at 25 to 27 degrees. But when you get that cool weather, the temperature goes down around that 20 to 22, those varieties start to become more susceptible. And so it's really difficult to make decisions about going back into cotton when you have such when you have this aspect to deal with, because you can't forecast that environment terribly well, and then you've got a variety that is changing its virulence based on that environment. It's a very difficult situation. Here is some data from Stephen Allen, and it just clearly shows you how the environment influences disease incidence. So all of these, here is the incidence from 0 to 100, and then we have the rainy days, we have radiation, less than 20, and this graph shows you relative humidity above 90. So just what I basically wanted to show from this data is just to see the incidence is in the red and the other term is in black, and that the pathogen, the incidence seen in the field measured, follows the number of rainy days or the radiation less than 20 and the relative humidity. just to show you just what such an impact the environment has on the development of disease. And human activity was that last one that has a great influence on when something becomes widespread. And this is no criticism of what anyone does. This is just the fact that human activity in agriculture has an impact on the disease triangle. And it's just something to think about, that everything that we decide to do potentially will impact that disease development. So, for example, whether you grow a monoculture, what your variety choice is, what nutrition you decide to choose, the rates that you decide to apply, your irrigation, residue management, weed and volunteer management, the permanent beds, constantly placing into where the inoculum will be highest. And then something that I think we often overlook is just farm hygiene and how much we're spreading that pathogen around. So no criticism on human activity, but just to highlight that, gosh, everything we do potentially and probably does impact that ability of that pathogen to develop and cause problems. So I've been asked just to briefly talk about all those management strategies and what part of the life cycle is actually being impacted. So when we look at all of these, all of them actually are impacting, either they're eliminating microsclerotia or they're slowing the build-up of disease, slowing the build-up of the inoculum. So all of these management strategies are directed to do this. And I just wanted to point out here about nutrition, because there was a question that came through about nitrogen. And I just wanted to mention about, because we talk about balanced crop nutrition, especially nitrogen and potassium, but there is research that shows that, so in this research I've done overseas, with added N and added K, the root exudates reduced microsclerotia germination and disease progress. When they removed the K or lowered the K, there was an increase in the germination of microsclerotia in the soil and an increase in disease. So when we talk about a balanced crop nutrition, this is what we're talking about. It's not necessarily whether you have high N, it's about that balance of N and K probably. And also all your other nutrients. There's many nutrients that have a role in the defence of plants to disease. But I just wanted to highlight just some of this research, and that there's a lot of work being done overseas, and they're actually now understanding what actually is the impact of those nutrients, and they're showing it's in the root exudates coming from the host plant, and therefore how important actually that balance is. And of course, always practice good farm hygiene, and this is all about minimising the spread of inoculum. So what is the effect of the pathogen inoculum on disease development? This is some work from our rotation trial at Gedda, and I just wanted to give you just data from cotton fields, not overseas data, but from our cotton fields with our strains of Verticillium. We are showing, so in this trial, it was a 3-year trial where you had 2 years of either sorghum, corn or a fallow, and then we went back in the 3rd year into cotton. And so the disease incidence was measured in the 3rd year of cotton over time. And then we also looked at what the inoculum was measured as picograms of DNA per gram of soil at the end of that season. And so what this showed was that, yes, the disease inoculum, there is a relationship to infected plants. We know that we can slow down the development if we have lower inoculum levels. And also that rotation crops such as sorghum and corn, and also fallow, but I'll talk about that next. But these crops reduce inoculum. They reduce disease incidence and reduce disease severity. However, their ability to have an impact on inoculum depends on the level that you start with. This is a very high inoculum field, so you may need more than 2 years out of the cotton to have any benefit. So this is because I mentioned about how like the bare fallow reduced inoculum, but I want to just stress that with our work with Gupta, we're looking at soil health. And I want to stress that even though, yes, the Verticillium dahlia inoculum is being reduced, but we're trying to understand, well, what impact does that have on the microbial composition and abundance in the soil? How does all these rotations, how do they impact the ability of a soil to be suppressive to disease? So in that trial, Gupta had soil samples and he measured abundance of fungi and microbial catabolic diversity, and these are indicators of the activity and the diversity of microbial organisms in the soil and how they've been influenced by the different carbon sources that have been put in. So from the work, we believe that to promote disease suppression, because we know it's in the literature that, yes, you need to promote disease suppressiveness, you need to have higher fungal diversity. So we have shown that with corn and sorghum, if they are in your rotation, they have a significantly higher fungal diversity and abundance. So maybe a bare fallow is not the best choice, even though it reduces inoculum, but it also reduces significantly all those other things that we feel are really important and know to be important for disease suppression. So we've also looked at looking for what are the hosts, and in the literature that CottonInfo produced, we certainly have like the updated rotation host table, which is really good information as to what we know to be hosts and non-hosts and asymptomatic hosts. But I just wanted to show you that we're looking at a lot of crops in the glass house to see whether they are hosts or not. But this also needs to be verified in the field. But just based on the work so far, certainly grain and forage sorghums are non-hosts. Millet is looking to be a really poor host, so a good option. And corn, even though it's been recorded as an asymptomatic host, it's a really good option for bringing down inoculum. The other thing is weed hosts. There are many, many, many weed hosts for Verticillium dahlia. And like Karen's been working on Agura burr and isolated the different VCGs. And so this is something that must be managed in the field. If it's there, it really has potential to spread the pathogen around. The other key one in our fields for cotton is cow vine, peach vine. That's a host. And even though not listed in this table, fleabane is an issue, but I've read that it's also reported as a host of Verticillium dahlia. So there are 3 weeds that would definitely be key for managing. So residues, why? So in the residue, that hosts all sorts of pathogens, not just Verticillium, but but many of them. And if it's undecomposed, it then is an inoculum source. And this is an example of Alternaria early season, where we've got all of this trash on the ground. The seeds have germinated through this trash and then they've become infected with Alternaria. But if we're able to get that trash into the soil early enough and have it broken down, then hopefully, and we shouldn't see these issues. A key thing though is that breakdown of inoculum of the trash and inoculum requires moisture. And we saw that through the drought. And that's why we have had a lot of carryover of the trash. Without soil moisture, the microbial activity is not going to be there, and then therefore you don't get the breakdown of your trash, and you don't get the breakdown of the Microsclerotia either. Someone was asking that there was a fellow asking a question about I think adding urea or adding fertilizer in aiding crop residue. Certainly, I've read that if you have a large carbon to nitrogen ratio, by adding urea, but with moisture, you have to have moisture, that this can aid and encourage breakdown. So that's probably something that would be really good for us to talk about. And so when do we need to manage? Well, vert, you need to incorporate as soon as possible, and that is to get it broken down, get the Microsclerotia, get all that activity, get everything broken down as soon as you can to reduce the inoculum as best you can and as soon as you can. Fusarium is different. We leave that on the soil surface and we solarise it. And this is 140-day solarisation. And that's because it's a really good saprophyte and it grows on residues really well. So we want to solarise that and kill as much of the pathogen to start with before we can then go into the soil. And with other crops, you need to bury them or bale them as soon as possible. Farm hygiene, why? Because you can move that pathogen around in soil, in water, anything that is contaminated with the pathogen, you can, and if you're moving that around, you move the pathogens around, but also weeds and other diseases. And the key thing is, once you have that in the soil, once you have verticillium in your soil, you can't eradicate it. And so it's really important to just try and prevent movement. When do you conduct farm hygiene? Always. And these are the things that you can do, and that's with the wash down machinery, clean boots, well-signed areas in your farm, inform people, talk about it. I think if we just talk about it and really just focus how important it is, it's something that everyone can do, and it really does help reduce movement of the pathogen. Probably talked way too long. I'm sorry about that, but thank you for listening. That's my summary.
Thank you, Linda, for your extensive introduction into VERT. You've saved me quite a bit of time. There's a few slides I will be able to skip over quite quickly because you've already covered those areas. And I will run through Verticillium, the work I have done using CeroX product, and also try and answer a few of the grower questions that we have coming up. So I don't really need to spend a lot of time on the terminology because Linda has already been through that. So I will just skip past this slide. What I will do is spend a little bit of time on the pathotypes. Now, this is a slide from the results of our PhD student Pearl when she was looking at the different pathotypes and different VCGs and the different virulence. What I would like to point out in this slide is on the left-hand side, we have the molecular typing. On the right-hand side, we've got images of the plants that were conducted, and this is a glasshouse trial. So we know, as Linda has already pointed out, we have in Australia defoliating pathotype, and these are usually described as virulent, especially in other countries. In Australia, we also have non-defoliating pathotype, less virulent, and these are VCG2A and 4B. So here you can see quite healthy plants. And we also have another set or cluster of isolates that come back as non-defoliating pathotype. However, in Australia, we see them actually defoliate and kill the plants. So these are quite virulent. I don't really need to go through the life cycle because Linda has already done that. However, what I would like to point out is when we're looking at the disease management, when I was looking at conducting the field trials for X, it was concentrating on this section here where microsclerotia are actually formed in the dying and dead tissue. So once again, concentrating on when microsclerotia are formed. So it's as early as just before December through to February. And the important point here is we're trying to reduce the density of the viable microsclerotia in the soil by targeting the life cycle stage when these microsclerotia are being developed. And the key point here is before they're returned to the soil in the dead and dying tissue. I just wanted to go through some of the really important points, I think, about microsclerotia. Now, each of these microsclerotia are composed of compact masses of pigmented cells. If you look at the diagram on the top, you can see quite a large grouping of cells. These are dark in pigmentation. They contain food reserves, and this actually enables the pathogen to survive, as Linda had already pointed out, for more than 14 years. This question comes up quite often, so I've included the reference here for people if they want to read more about how they came about determining that status of 14 years or more. And I would also like to stress the point that these can survive in the soils for more than 14 years without a host. They're very, very resistant to harsh conditions, which of course adds to their survival. The highest germination is between around that 20 degrees Celsius and a pH of 8. However, this does move up and down, and they're quite capable of germinating in lower temperatures or pH. Now, these single microsclerotia, we're calling them a group of cells, can germinate up to 13 times. And the number of these germination events has been correlated with the microsclerotia size. That's a really important point, I think, for future research, is to look at the impact of the size, pathotypes, and virulence that these actual survival structures have. Given that a large microsclerotia may have a higher DNA content, it may have more opportunity to germinate several times. So if events are not conducive, it might actually add to how it prolongs in the soil. I've also included the reference here for people to look into that more. Now, germination will depend on the availability of free water and, as Linda had also pointed out, stimulated by exudates from the root hosts and non-hosts. So where did my work begin with X? Back in 2015-16, I was looking at products because we always screen for anything that may have an effect on Verticillium. I want to point out that this does inhibit the development of microsclerotia. At no point will I ever say it is killing microsclerotia. If you look very carefully at these Petri dishes, they're containing Verticillium isolates. Each of these are 4 millimeter diameter fungal plugs. Surrounding those are 4 millimeter wells or holes that have been taken out by a cork borer. X was added to these or not, in this case on the top left-hand side. This is the control with just water. Then we use 2 litres per hectare rate, 4 and 8. What is absolutely obvious is the product does inhibit the development of microsclerotia. You can see it doesn't kill it. It still continues to grow around between where the product has been applied, but it does not develop inside and the margins around the wells. I must admit this was the most exciting thing I'd ever done when I walked into a the lab, opened up the incubator doors, and I was very, very happy. I've been screening things for many, many years and had no effect up until this point. So from there, we moved to field experiments. But before I go through those experiments, I want to just, or the results, I want to just run through what actually happens in a field in Australia. There's a lot of data overseas that we can all get hold of if you do your searches. However, I wanted to actually document what was happening in Australian cotton fields with Australian cotton or Verticillium isolates. You can see in this graph, this is around the planting time, and the inoculum is sitting quite low here, propagules per gram. It actually decreases naturally, and this occurs due to the Microsclerotia germinating, so there's less, and that's from the soil and root exudates, the soil moisture, so it will naturally decrease as the season progresses. However, after defoliation and incorporation, we see a massive jump in the amount of inoculum measured as propagules per gram returned to the soil. So when I thought about targeting on applying the products, I looked at when the first symptoms were seen in cotton fields, when they were obvious, usually around that mid-February, early March, and then towards the end after defoliation. So that's how I chose the 3 application times. So the field trials were conducted over 2 seasons in 2 regions with 5 fields. I've selected 2 sets of data from 2017-18. The graph on the top represents a field with very low incidence and also low propagules. Now in the blue is no foliar applied, no product. On the red is where the 0x was applied, and this occurs for both fields. I guess what I want you to see here, or perhaps you can see it yourself, is that where the treatment was applied, we had a decrease in the microsclerotia, the measurement of propagules per gram in the soil, as you can see on the graph on the right-hand side. If you look at the left, where no product was applied, the microsclerotia actually increased. This was consistent across our 5 experiments and the 2 seasons. So then 0x actually was registered for the management of Verticillium wilt. Now to undergo that, all of the information data was sent to and tabulated and sent to the APVMA for them to consider a label change. The work continues with Innovate Ag and also Crown Analytical, quantifying inoculum in the cotton fields. Thankfully, there's a DNA way to do it now, which is much, much quicker. It's a fast turnaround, high throughput, much less labour intensive. And I do believe it requires much more investment in research so that the full benefit of getting a quantitative, fast measurement of inoculum in the soil can be available to growers and consultants. The idea of having a quantified amount means you can measure. What are we going to measure? The effect of any rotations, the effect of any products applied, the effect of weed-infested areas. The ability to measure gives you an opportunity to see any change. I'd also like to inform people that we are actually undergoing an ARC linkage grant. We're successful in that, and we'll be looking at determining the mode of action with 0x on Verticillium dahliae. Disease management, Linda Smith went through quite a lot of this. I think I'd just point out the really important point here is to incorporate your stubble as soon as possible if you know you have Verticillium wilt, and retain the stubble on the surface for as long as you can, at least a minimum of 1 month if you have Fusarium wilt. There's some key publications, type in Verticillium wilt Australia. This is just 3, but there's so much, and also CottonInfo is your source for most of the up-to-date information available for growers. Linda also touched on the weed management. This graph was taken from the work that Peter Lonergan and I reviewed. I think it was back in 2015 in the old weed pack. So if you have a look at that, but the important point from this table is there's 36 weed hosts for Verticillium. 8 of them host more than one pathogen. So we're looking at Verticillium and or Fusarium, or it might be a host for Verticillium and black root rot diseases to occur in your fields. My top 3 would be Belvine, for the reason that it actually is a host for 3 separate pathogens. The same with cotton. Don't forget your regrowth. Please, please control that. And I'll touch on Nagura burr. I think this is a really important weed that growers should do their best to control. As Linda said, I've been doing some work with Nagura burr. Now, the average number of Nagura burrs per plant is around 144. Please have a look at this photograph on the bottom right. This is inside a storage dam on a cotton farm. I don't know that I need to really emphasize how many plants are inside this storage dam. So, getting back to the average number of burrs that are infected with Verticillium, it's around 40% of these plants. The number of Microsclerotia per burr is more than 2,000. So therefore, if you extrapolate that out, the number of Microsclerotia per Nagura burr can be more than 115,200. Now, the number of Microsclerotia per cotton plant is in excess of 250,000. Just under half of that is in Nagura burr. So when you see one Nagura burr plant, please try and target it and remove it. Also, don't forget the impact of the pathogen being transported in your water. The number of Microsclerotia per litre of dam water is 15,000. Now, we do know that Nagura burr is a host to VCG 1A and also 2B, but I have just submitted a first report of VCG 6 in Australian Nagura burr. I can confirm it does infect cotton, but I have not done pathogenicity testing, and the impacts of this new VCG in the cotton growing systems remains unclear at this stage. My key messages for you. Verticillium dahliae is a highly adaptive pathogen. New VCGs are being discovered. As Linda said, prior to 2014, we only thought we had VCG4B present in Australia. In 2014, Linda reported the first report of VCG2A. In 2016, my colleague and I reported the first report of VCG1A. And this year, we will have the first report of VCG6 in Nagura burr. This disease requires very long-term management. Microsclerotia can survive for more than 14 years with no host. There's no one-step approach to controlling it. 0X, in my opinion, is just one more strategy. Control your alternative weeds and also your cotton. I'd like to acknowledge this work as a compilation of many projects over the years. I've got a few slides where I've tried to answer a few questions. So the first one was the Microsclerotia. Both Linda and I have touched on that, and the reference is there for you all to have. It should be saved. The next question that I received was, can durum wheat carry host vert? Is it a good rotation? Yes, it is a very good rotation, in my opinion. I did some work from 2013 to 18 looking at cotton rotation trials, including durum wheat. So if you look at the graph on the left-hand side, these are 6 GPS sites that we went to every single year. So we have 6 different sites indicated here by various colours. Where there's none, there was no propagules measured at that time. So very early, May 2013, following a cotton, the propagules, look at the numbers, there were more than 900 propagules per gram of soil. So following durum wheat, the propagules declined, and then again following sorghum continued to decline, and it looks like it's 0 all the way along. So after 2017, so if you jump to the graph on the right hand side, I've reduced the bar graph on the left hand side, the measurement, so you can see that after sorghum we had much less microsclerotia in the soil. Again, following durum wheat, it decreased again. And then following cotton, it started to increase again. Now, all of the literature, whether it's in Australia or overseas, will tell you that once you plant cotton, it's a great host. It will increase your inoculum in the soil. I thought it'd be really important to let people know there's some brilliant experts in Texas looking at Verticillium in cotton. Dr. Jason Woodward, he's done a lot of work with rotation and inoculum levels. I've got a few references here. The main point about this was the cotton rotations and irrigation. They looked at 3 types of irrigation: their normal, a reduced irrigation, and then a larger irrigation. When you see cotton rotation, each of those are high. I guess the picture here is that the sorghum cotton reduces it dramatically compared to the cotton. And again, the sorghum cotton starts to increase again. Again, looking at rotation, the effect of irrigation on that, this is just amplifying that first slide. So you can see the reduction, and the impact of irrigation here is important. When you talk about disease management, people often ask, well, what can I do with my rotation crop? And my advice is don't just look at rotation, look at the effect of irrigation and your rotation on that. Bit more work on irrigation. The temporal dynamics of microsclerotia. They are affected by the irrigation amount. Again, here we have the low, which is indicated by a light blue colour, the red, which is a moderate irrigation, and the black is a high irrigation. Now, in 2011, this was a very bad year for drought, and so more irrigation was applied. But I guess you can all see that it increases with the higher irrigation compared to low irrigation. So irrigation in itself does affect the microsclerotia in the soil. Further work by Jason was looking where they had they were trying to keep up the evapotranspiration by 80%, but that only went up to 2009. And in 2010, they were in keeping with 60% of the water requirements and looking at the actual incidence of verticillium, not the inoculum level. We've got high water, and this is cotton, lower high water with a rotation, baseline cotton, and baseline rotation. So when you see the dashed on the green here, you can see it is much lower at a base rate with a rotation compared to a high cotton. And I think that's a really important point that people should think about. Don't just think about your rotation, think about the impact of the moisture content in the soil. Nitrogen, again, what's the impact of high nitrogen? Well, in some papers, wilts induce more rapidly in nitrogen-rich soils compared to nitrogen-deficient soils. Also, the ability of starved isolates, this is the Verticillium dahliae isolate, to penetrate a host is diminished when they're in a nitrogen deficient or less than optimal for the pathogen, not the plant. Here's another reference that you might be interested in reading, the effects of nitrogen supply. This is not in cotton, but the microsclerotia develop and behave very similarly across many hosts. And that's all I have for you. Thank you so much for listening.
Right, I'm just going to give you a bit of an update from a disease management point of view. We've talked about some of the things, obviously, with Linda and Karen's talks. A lot of this is based on the stuff that we've actually probably shown quite a few people with our cotton management tool, but we do have some up-to-date data that we've added into that, and obviously in a month or two's time we'll have a lot more information once we get through picking. Just on the actual variety V ranks and F ranks of the diseases, I've just got a couple of photos there that you've probably already seen, but certainly verticillium wilt, common look of it is that speckling, mottling of the actual stem once you cut it, while the fusarium wilt is actually that red sort of ring that you actually see splotched across the actual cortex. And obviously the scoring system for fusarium goes from noughts and ones, being less infected, and then rated against 2s, 3s and 4s when we do the F rank, while verticillium wilt is basically with V rank is just presence and absence. So the V ranks, we have a new variety, which some of you might be aware of, which is called Psychot 606 B3F. It's been in our V rank trials for a number of years now. We have 19 trials of that. And certainly we've seen a significant difference between Psychot 606 B3F compared to the standard, which is Psychot 714 B3F, which is a pleasing result. Certainly when you walk across these trials, which are in planter width by the length of the field, quite numerous reps, up to 20 odd reps. You can nearly pick the 606 across a lot of these plots because it's quite evident that there's less infection in the main stems with this variety. I've just mentioned the F ranks there. No, it's not necessarily that pertinent to today, but certainly we do have some really strong, quite resistant varieties. And 606, unfortunately, we've only got one trial there, but at this stage it looks like it's similar to 746 and 748 in terms of its resistance to fusarium. So verticillium wilt management, it is a balance between acceptable disease presence and economic sustainability. As we mentioned before, it's not going to go away. It is in the soil. And I guess farmers need to work out what disease they have in their fields and what they are going to do about it. But there is options. Some of the work that Crown Analytical done, which we've mentioned over the last couple of talks, is certainly allowing us to look at the disease from a different perspective, from a field-to-field basis and an area-to-area basis. And you can see some of the different inoculant levels in these three fields here. And certainly you would pick a variety that would be more resistant to the right and middle, and you could probably go your 748s, your 746s certainly in the field to your left. So it is very, very important in terms of variety selection, and typically what we see is that in medium to heavy soils of the disease, we certainly see the most resistant or tolerant varieties go to the top in terms of bars per hectare. Certainly 714, 707 and obviously Psychot 771 BRF are not around these days, but certainly in those trials they did stand out when we had heavy disease. And I've got an example in the next slide of 606 where at CSD we've got a CSD management trial that we put in every year. And in the blue, you've got infection levels across those different treatments. And in the actual orange line is actually the yield. So at the end of the day, 606 B3F, it had the less infection in the stems, and also it had the most yield under pretty moderate disease pressure. Now, Karen's just put this trial data up, and her trial data and the way she displays is a lot better than mine. But I guess it just says from a crop rotation point of view, it's very, very important to look at the other crops. Certainly, Linda and Karen have actually expressed that you can actually reduce those inoculant levels right back to zero. Once again, when you actually add cotton back into that rotation, it rises. Now, one thing to note with Karen's data, and this is Karen's data, is that they went back to a variety that was not necessarily tolerant to the disease. So even though the levels were fairly low, they went back and grew a variety that didn't have a good V rank the following year, and hence probably one of the reasons why it started to go back up. So some of the work we've done in terms of temperatures, we've looked at skip row and higher beds, and the graph on the actual left-hand side is showing temperatures of soil, while skip row, single skip on the right-hand side, and certainly the skip row had warmer soils pretty much throughout the season. Those warmer conditions obviously help with the disease. The warmer it is, the less disease, I guess, virulence. Now, this is some of the work that Steve Allen's done over a number of years in terms of irrigation frequency, and it backs up and supports stuff that Karen put up there. But cooler climates do induce the perfect conditions for verticillium wilt. However, low irrigation frequency means we see less verticillium wilt incidents. And we do see this at Breezer where they're only irrigating once or twice a year, up to 3 irrigations. It just doesn't seem the severity or the incidence of the disease as much as what we see where we're irrigating up to 10 times. Certainly overhead irrigation with reduced root system structure and high irrigation frequency is the perfect scenario for verticillium wilt. So we're irrigating every sort of 5 to 6 days, up to 7 days. We're putting on a certain amount of moisture. It's cooling the soil down, and causing significant issues around, particularly the upper Namoi, with this disease under those sorts of irrigation systems. And in dryland crops, we don't see it, and that sort of backs up and suggests that obviously irrigation and the amount of water you're putting on can actually cause significant issues in terms of the amount of verticillium wilt in irrigation. There's also some work that was done by Steve a while back, and what we've got here is on the table on the left-hand side. We've got a lateral move field and also a furrow irrigation field. And right down the bottom where it says total, you've got the grams of root structure. So 3.85 grams on the lateral move field versus 1.96 on the furrow irrigation fields. Now that's from dry root weights, 0 to 15 centimetres in that thing. So you've actually got a lot more massive roots basically under the lateral, right, at that 0 to 15 centimetre zone versus the actual furrow irrigation at 1.96 where it's probably going deeper. Now the graph on the actual right-hand side shows, and this goes way, way back, probably near 15 to 20 years back, where the furrow irrigations are the white squares and the actual red diamonds are the actual the overhead irrigation. And certainly the infection or the actual incidence of disease is higher in those lateral fields. We talked about nitrogen rates, and this is just a trial that we did for a 2 to 3 year period where we looked at various treatments right across the board. We also had nitrogen, and the nitrogen rates of over 300 kilos per hectare can potentially have high yield. We've seen that, but can result in high incidence of the disease. And certainly in this trial, we saw that it was the highest yielding at 11.99, but it also had one of the highest incidence of the disease. So you've got these large, plants that potentially have a larger spore load going back into the soil, which is creating a problem for the following year. So you really need to have a balanced nutrition program and set yield targets and apply nutrition based on target yield. Plant population. We've done trials over a 3-year period now. This stems from work that was done at Texas A&M, where they looked at the actual incidence of disease using a high plant population. Certainly we saw, that a higher plant population had less infection levels compared to the farmer's standard rate, which was lower, 52 to 46%. We tried to establish around about 19 plants versus 13 for the lower rate. For some reason, we weren't getting the separation as much with the high rate compared to the actual farmer rate at 9.1 and around about 12 plants. In saying that, we were seeing quite a few dead plants across these trials over the years that had died a lot earlier than expected. But overall, what it means that even with the lower population, you got less plants that aren't infected. When you actually spread that over the field with a high population, your ratio basically means that you've probably got more plants that are actually diseased in the paddock at this point of time that are potentially lifting your actual spore loads. And there was no yield difference basically between those trials statistically over the 18 trials having a higher rate versus a lower rate. So we talked about reducing inoculant sources, and certainly, you know, the greatest number of Microsclerotia are formed in the leaves, fewest in the roots, but most Microsclerotia serve as inoculum are formed in undecomposed residue. So it's very, very important, obviously, to get onto it quite quickly, plough them back into the ground, break down, you know, that Microsclerotia that's living on the leaf material, let it rot as quickly as possible. And obviously you do need moisture, as Karen mentioned. And they can survive in the soil, as we've found out for many, many years, unfortunately. So Karen's actually put that graph up previous to this. It's basically showing, obviously, you know, that you do get natural attrition of the disease over a period of time throughout the season. But as soon as we go into defoliation mode, you're actually putting that straight back into the actual soil. That's why it's very important to try and break it down as quick as possible. So planting date is very, very important. You know, we need to manage for earliness with this disease. Typically what we see is that when it does under bowl load pressure at the end of January and into February, if you do see those drop off in temperatures, you know, there can be a spike in the actual disease. We're heavily irrigating through this period too. And if you do get that spike, you know, sorry, in terms of the drop off in temperature and the spike in the disease, we can lose those 6 to 8 nodes on the top of the plant, which dramatically actually can affect the actual yield. So, you know, trying to get the crop in as early as possible without causing delays due to cold stress is, you know, it's not easy, but certainly creating an early crop with this disease is very, very important. At one stage, we're trying to get 4 bars per acre. These days, sorry, 10 bars per hectare. These days, we're trying to get 15 bars per hectare, which has meant that basically we're extending the season in many circumstances, trying to capture those top bowls. But in disease fields, this is certainly not the go, because you can actually suffer greater losses by doing that. And just as a summary, you know, you need to know your disease inoculum levels, certainly the crown work that's being done at the moment, and understanding from a year-to-year basis what you've got in the ground, and then cross-checking that against your management is very, very important. Selecting the most resistant variety is also very important in terms of using 606 and Psychot 714 if you've got medium to high disease. Having a well-developed and fine-tilled fields that are preferably higher than normal, increasing the soil temperature is also important. We saw that with the skip row work. Optimise nitrogen levels and do not over-fertilise. Plant as early as possible to develop an early maturing plant. Getting it in and getting it off is very, very important. Pre-irrigate rather than water up is more preferable. We don't want to be putting cold water, particularly in areas that are already, you know, cool during that sort of first couple of weeks of October, can actually bring on the disease. And we've sort of seen that this season, unfortunately, particularly in the Upper Namoi, where we had a cold start and with cold water going on plants. Develop a root system that penetrates below 30 centimetres on both furrow and overhead irrigation. And this is certainly going to be an issue with overhead irrigation, but I think first irrigation is very, very important in terms of trying to get that root system down as far as possible before we actually, you know, have to start irrigating. And utilise products in the season that can decrease the amount of Microsclerotia going back into the soil. Karen's talked about 0X. And having a crop rotation with non-hosts of Verticillium wilt. So certainly, you know, the guys that have used some of the other crops in rotation, particularly, you know, corn and sorghum, have seen drop-offs in their disease on their properties that once used to be chock-a-block full of disease. So it's certainly worth rotating those. And sort of come clean, go clean is very, very important. You know, we've probably forgotten about it a little bit in terms of, you know, making sure that you're coming onto properties with clean cars, you know, and basically clean shoes. And remember, it is a balance between acceptable disease presence and economic sustainability. We've got to live with this, but certainly we can still get reasonable yields and pretty good yields. And it's just not one shot, you know, to cure this one. It's actually an amount of using different management practices to make the crop as efficient as possible under the disease incidence. And I think that's me.
Thanks, Bob. So we will open the floor to questions now. I can see there's a bit of discussion happening in the chat function. So the impact of raking and burning. Karen, did you want to say anything there, or do you want me to read out what you've written? No significant difference because raking distributes the microsclerotia throughout the field, but perhaps this should be looked at again with the use of 0x, as this may lower microsclerotia in leaf material returning to the soil. That's a good point from a question that we did have emailed in beforehand.
Does anyone need any more info on that, or we'll just move on?
Oh, Janelle?
Yeah, Karen, so the raking will spread it. Does the burning kill it?
We didn't look at the effect by itself. It was a trial where we had raking, burning and nothing, and we did measurements prior to and after. I do believe that the microsclerotia can withstand very, very high temperatures. It would depend, because it's usually not a very hot burn when you're spreading it across, you know, in a field. We didn't get a long lasting hot burn, but I mean, have a look at it.
There's another one there from Craig. Can flooding of a field for a few months or longer reduce pathogen levels? Hasn't been looked at. I think it's been looked at for black root rot, but maybe not in terms of vert. So that's something that we could potentially look at.
I haven't done anything with that. I'm not sure about anyone else.
Were there any other questions from anybody still on?
Yeah, it's Craig McDonald, Narromine with CSD. Just seem to remember a few years ago there was talk of actually flooding fields, like obviously irrigation fields with banks around them, to possibly control the pathogen levels. Is that still any more work being done on that, or is that still an option? Or I guess we've had many years of drought. We've had a few years now with some water. Might be an option in some smaller fields, perhaps.
As far as I know, the work on flooding in the south was concentrated for Phalaviopsis basicola, the pathogen causing black root rot. I'm not aware of people trialling it for any effect on Microsclerotia for Verticillium.
Okay.
I mean, it's been done for Fusarium, but I haven't. Yeah,
Remember
I don't
that.
know that.
Just curious about Dahlia. Yeah.
Yeah. Oh, Karen, do you know if a non-host stimulates germination of Microsclerotia to the same extent as a host?
Yeah, I do know that they can, because it's the actual root exudates of the plant that's stimulating. We're not talking, there's two different things, germination and infection. So if you put a non-host in there, and hopefully under the right conditions, moisture and the exudates from the plant, it will induce germination. That's completely different to germinating and infecting a host.
Yeah, so is it, but it's to the same degree, the exudates. So if you have a non-host, so I'm just thinking, like, if you had a non-host and it doesn't get infected, but you could stimulate germination of Microsclerotia, could you, are you reducing then the population?
Yeah, and that's particularly how crop rotation works. It's not the actual crop that you're putting in, and that's why all of the different crop dough rotations actually have an effect if they're in reducing the inoculum level. So it's a combination of the exudates from the plant, also the moisture level in the soil for the pathogen. There is a paper, and I read it many years ago, and it's I can't remember who to reference. I can't find it again, but maybe someone smarter than me can go back and check. But I've had it written on my whiteboard here for nearly, I don't know, 8 years, and it says Verticillium vascular infection success is estimated to be 0.02 to 0.06% of root infections in cotton. So this is why it's really hard when you're looking at inoculum levels. You're not just measuring thresholds, because they're going to be an indication of risk. So you might have, I don't know, 20 microsclerotia or propagules per gram, but when you bring it back to the rate of success of infection, can be why you can have a field with, I don't know, maybe we should be talking in grams, but I only know the propagules per gram. So I could have fields with maybe 60, 70 propagules per gram, but really low infection, like the incidence was low. So it's very difficult to get a threshold that's completely accurate. And I would urge that any disease matrix, we were working on one and gave a really good indication. And that's why I only ever quote the minimum disease incidence, because of that infection rate or the success of infection can change so much.
Yeah, sorry, yeah, I just was just interested to see if there's a difference in the exudates between a host and a non-host and the stimulation to germinate, but yeah.
There is a really good researcher, I'll try and think of her name. She's American at Charles Sturt University, works on exudates of plants and weeds. So maybe I'll try and think of her name.
Thanks. Yeah, that'd be interesting.
I've got another emailed question here. The use of products such as cloudy ammonia, bleach, or liquid urea as sterilants. So that's aiming it after mulching. Can you spray those products at a high rate, hoping to sterilize the trash that's still there?
I guess I'd be concerned about.
I'd be concerned about everything else in the field.
Yeah. And
I think
don't know how effective that would be.
Yeah, I mean, using the word sterilant for disease management, I would steer away from, because if you're sterilizing, you're going to kill your beneficial microbes as well.
Yeah.
And a lot of those, a lot of the inoculum is actually embedded as well in organic matter, so it would probably be very difficult to get penetration into all of that material for effective treatment.
You might end up with just vert.
Yes, you might. Yeah, I think it would be very difficult.
Yeah.
Beth, do you know, Montgomery, I've just got a question for Rob Long. I could see him on there. So if Rob, if we're looking at mapping our verticillium levels or inoculum levels in the paddock, like to get a bit of a baseline of where we're at now, like are we looking at newer fields or like how do we use those baselines for benchmarking?
Well, there's actually the opportunity now to go into fields that have, you might see variability and you're suspicious of verticillium. So you can actually go and get a point in time post picking. The best benchmark, of course, is pre-plant, because that's when, that's a predictive tool. So you've got an idea of you're taking samples from exactly in the plant line, and by that time the beds are pulled up, and you just go and create an inoculum map based on that testing, and it's your reference point to come back to for when you want to plant cotton again you know, in whether it's back to back the next year or 2 years' time, or if it's severe, you want to give it a 3-year spell, and you do all sorts of other management strategies in the meantime. You can come back to exactly those same sites and get a very good and accurate measure on whether your management strategies have been a positive or a negative. There's no real formula as to whether you start on a dirty paddock or a clean paddock. If it's dirty, then if it's a dirty field, then you just know that you want to reduce that inoculum as best you can with best management practice strategies. If it's a clean field, and we've got that, we've been testing fields that have had 20, 25 cotton crops in the last 35 years, and we struggle to find any inoculum, any verticillium in those fields. What is it about those fields? So it's really what answer that you are striving towards as to where you start, Janelle. I'm not sure if that answers it properly, but it's my best indication. And I just sort of use the analogy of you need the numbers. There's a tool there for DNA assessment that is reliable and repeatable, and it's I just use the analogy if growers and consultants are professional about it, do you want to be an athlete who really measures your performance well, or do you just want to be a jogger who just goes for a run? Knowing the numbers, Kathy Freeman wouldn't have just gone for a jog in the lead up to her 400. She would have been measuring herself and monitoring her performance seriously. I see a great analogy here. If you're really serious about monitoring your performance in the inoculum, you just need to use the measurement as a baseline, as a strategy to monitor your what you're using.
I think the bench, knowing where you're starting, Rob, and Karen, you might comment on this in terms of the use of 0x. If people are considering using 0x, we know it's not going to get rid of the issue, but it's going to reduce the increase of inoculum levels. So, is it it's good to kind of know where you're sitting before you use that product, or?
Oh, look, I think the industry has to start, like Rob said, we've got to stop just measuring the effect of verticillium in crops. We know it's bad and there's different pathotypes, but for us to have any meaningful impact on reducing losses associated, you need to look at the cause, and that's inoculum. And then you can start with measurement, means you can start applying different products, different strategies, and actually quantify what impact it's actually having, whether it's 0x or a combination. And I'd really like people to sort of think about it as the old-fashioned best management practices and not single out one. Of course, growers will be able to pick the strategies that are appropriate for their individual farming practices, but I don't think we should be saying measure this and just do this. It should be tailored to a best management practice with many strategies available to growers, and they choose the ones that are suitable for their farms. Of course, I think you should definitely measure before, during, and after.
And the one thing that the measurement, it does empower people or growers to run case studies on their own farms. They can, if they know what the benchmark starting numbers are, then that the pathogens can be differentiated between work out whether it's defoliating or non-defoliating. The CSIRO have developed that so that there is that differentiation now from this DNA test, and then Yeah, exactly as Karen said. I couldn't have said any better. You just monitor your own management practices and see whether it's working or not. And of course, the next extension of that is trials. Is it with nutrition? Is it with 0X? Is it a combination? Is it irrigation strategies? Is it flooding? All of these things. There are tools there to measure their impact. And like crown rot in wheat, it's a multifaceted management strategy to get a good result.
Thanks, Rob. Were there any other pressing questions? Okay, so I'll just quickly summarise what we've learnt from today. So for the growers still here, over the next few weeks, as we come to the end of the season, these are some of the things we can be doing to reduce the impact of Verticillium wilt on farm. So correctly identifying the disease means going out with a pair of secateurs and cutting stems and looking for that mottling. It's really important to make sure it is Verticillium wilt and not Fusarium wilt, because the management practices are very different for those two diseases. So we suggest keeping some secateurs and sample bags in the ute when you're driving around, so you can cut those stems. If you're unsure, take a sample of the stem and post it to probably Linda might be best. If you think might be Fusarium, definitely don't send those ones to Yui at Narrabri. The next thing you could do would be to consider using 0X with your defoliation. So it might be too late now in the season. I think the label, Nick, still has the 3 applications throughout the season. But Innovate Ag are doing some work looking at just that single application of defoliation. But whether that might help you going into your next season of cotton is something to definitely consider and take on board. And then your residue management. So as soon as your cotton's picked, try and mulch and incorporate that trash as soon as you can to get the breakdown happening faster. So that's right now for the end of the season. But looking forward, in terms of the whole season and your whole farming system, this is sort of a summary of management practices that you can use throughout the whole cycle. So in the off season there, after your end of season management, you want to be planting non-host crops. So some of the ones mentioned today have been grain and forage sorghum, some millet varieties and durum wheat. If you want some more information on rotation crops, the Australian Cotton Production Manual, the 2021 book has a table on page 78 and 79. I'm not sure the page numbers for the manual coming out this year, but that's available online and in hard copy. Control host weeds. So some of the other ones mentioned today, cow vine, peat vine, fleabane, bell vine, Mugura burr, and of course, cotton volunteers. Knowing your inoculum levels, so that could be getting in touch with Rob if you're concerned and getting that field mapping done through Crown Analytical. Then moving into your following cotton crop, you want to optimize your planting conditions. So have good fine tilth in your hills or beds, and you want increasing temperatures. So 25 to 27 degree air temperature, I think Linda was where we said the optimum was for ensuring your V ranks are working adequately. Selecting the most resistant varieties. So all that information is available on the CSD website, or your V and F ranks will be there. I think you do have to be a member to access that information, but it is all there online. Plant as early as possible while still avoiding the cooler conditions and black root rot if you're in a valley that has issues with black root rot. Optimizing your nitrogen levels and the ratios of nitrogen and potassium that was mentioned earlier. Developing a good root system, so over 30 centimetres deep. So you want to be drawing moisture from further down in a bit of a deficit irrigation at the start of the season. Avoid waterlogging and tailwater, and along with that would be to try and irrigate your least infected fields first, and then use that tailwater on infected fields rather than putting tailwater off infected fields onto clean ones. And then we're back to the end of season management. So IDing the disease and then considering other things. And throughout that whole system would be come clean, go clean. That's between farms, between your own farms and between your own fields if you have issues in some fields and not others. So were there any other questions before we wrap up for the day?
I think Yui's got his hand up.
Yep, Yui.
Yeah, I'm Yui here. Hello, everyone. Just because like you brought it up, Beth, for the application of like Xerox at defoliation, but we have to remember everyone we have both of the pathotype of Vidalia there, defoliating and non-defoliating pathotype. And lots of the crops that I went to this like season survey, they got defoliated already. So I don't know whether that type of recommendation at defoliation application is the best practice or not. So just consider what you got elsewhere, everyone. So
yeah, yes. So you'd have to be following the label directions for that. Obviously, if you defoliated, it's too late.
Yeah. So with the defoliating infection, defoliating pathotype infection, crops already defoliated. So probably the application of Xerox at defoliation is already late. That's what I want to say, because I think the defoliating pathotype been hanging out quite a bit this season. We've seen a lot of defoliated crops by far in the Namon. Yeah. So Karen, yeah.
You're still on mute, Karen.
A lot of the work that we did with the 0X was including fields with defoliating pathotypes. So we're not saying you're going to cover each plant, each leaf before it is defoliated. What we're saying, and it's all about the timing, and we didn't give dates, we said prior to, and it's when you start seeing those symptoms. That's your guide for, apart from at defoliation. That one's listed on the label, but that will come down to a bit of common sense. If your whole crop's already defoliated, you're not putting a defoliant on, are you?
I haven't seen that massive of,
yeah.
I haven't seen that massive defoliation of crops infected with like you know, so
yeah,
we've seen lots of defoliation, but like we
do have a lot of plants that, even including the fields with the defoliating strain, including the non-defoliating 2A that does defoliate, bear with me, that you'll get, you know, a lot of plants that don't have any external symptoms, but they still have the Canidia hyphae and the potential for the formation of microsclerotia within each of those infected plants. And that's what you're targeting. You're trying to reduce the formation of those in the tissues that are starting to die. But I would stress that growers use and follow the label rate. That's what the trials were based on. That's what the APVMA agreed to. But I'm not an expert on defoliation and the rest of, like, all I can say is follow the label rates, because that's what the research was based on. Going forward, it might change, and that's probably really good questions for future research. Like, you know, keep addressing the ideal application rates or timing of application.
Yeah, Janelle's just made a good point there in the chat. If anybody does have further questions or wants some more clarification around VERT, please reach out to your REO for whichever region you're based in. or one of the pathologists, and we'll get back to you with some answers. So I think that's probably a pretty good place to wrap it up. We have gone a little bit over time, so sorry for that, but I think we were having some good discussion in there, so I let it run. Yeah, so thank you everybody for joining, and I hope you've enjoyed this afternoon and have learnt something, and that we've clarified some of the management practices for dealing with dirt on your farm. And just a reminder as well that it has been recorded, this meeting, and it will be available on the CottonInfo YouTube channel. And I think we'll also make up a 1 or 2 pager summarising what we've gone through today. So that'll all be available on the CottonInfo website. So thank you, everybody, and thanks to our presenters.